All right, we're going to start today's lecture on the central dogma of molecular biology, where we discuss the different experiments used to identify DNA as the genetic material, and we're going to work on labeling the structural components of DNA. You guys know me. We talked way at the beginning of the semester about emergent properties, and we talked about the top-down view, reductivism. So I like to look at things from the bottom up. Start off with the chemistry, build a monomer, go from the monomer to a polymer up to an organelle to a cell to a tissue, tissues to organs to organ systems to organisms to populations to ecosystem. You start at the bottom, you work your way up. And if you understand the bottom, the top just makes sense. So we're starting at the bottom here. This lecture should tie together all of the loose threads from all the other lectures. We're going to learn today how DNA is replicated, how DNA is transcribed, and how RNA is translated. We're going to go from DNA to RNA to protein. This is the underlying process by which we get proteins. Back to chromosomes. You guys remember chromosomes, right? Yeah. No, don't remember chromosomes. Chromosomes are those things that live in the center of your cell. What's that thing in the center of the cell called? Nucleus. Thank you. Yes, the nucleus. Because if you didn't remember chromosomes, I was kind of iffy on if you knew the nucleus. With the chromosomes, you've got the structure of them in the center. They're bound at a centromere. There's a central point, the centromere, that the kinetochore surrounds. At the tips, you have the telomeres. What are chromosomes made of? Right, chromosomes are made of highly condensed chromatin. Chromatin itself is made up of DNA. DNA wrapped around histones. So far, so good. That DNA has a message. It's a polypeptide encoding region on it. What's that polypeptide encoding region known as? A gene. That's right. The polypeptide encoding region of DNA is called a gene. Those genes are going to be expressed as polypeptides. So first they're going to have that DNA on that genetic region is going to be transcribed into RNA, and then that RNA from that region is going to be translated into a polypeptide, which can then go on to form a protein. So I'm going to tell you fun stories. Science stories. This is the story of the discovery of DNA. These two guys are Watson and Crick. James Watson and Francis Crick. They were both British scientists who were puzzling over the structure of DNA. Prior to this, we'd found DNA. We'd extracted it by accident from the bandages of soldiers during World War I. We know what this stringy substance is, and we kind of were figuring out it's the genetic material. They were trying to put together the pieces. They knew exactly what elements were in it, what atoms were in it, but they had no idea how those atoms were put together. So they would sort of putz around their lab all day, not all day, but they would take, you guys have seen the what are they called? Kits where you have a ball and a stick, and you can build chemical structures with them, right? They might still use them in chemistry labs, I don't know. Or they're just like, put it into a computer, call it good. What these two guys would do is they take these balls and sticks representing different elements and different connections between the elements and start building models, trying to figure out how DNA worked. And they had a lot of weird shapes because it's almost like building a Lego set without having the instructions or a picture of what it's supposed to be. It's just a bunch of random blocks. They had no idea what they were doing. Everyone thinks they're really smart now. Turns out, though, they were just messing around in the lab. Have you guys heard of Watson and Crick? Some people have, some people haven't. They were very, very famous in the 1950s. Recently, they've come into some, especially Watson. Watson turns out he's a jerk. He wrote a book in the 1960s where it was all about the discovery of DNA, and he made himself out to be this brilliant scientist, this genius, and turns out he's not. But more than that, he had a lot of very 1960s-esque, for lack of a better word, views on women that he was very fond of putting in writing. And he still holds those views today and made people aware of it. Also, he has non-accepted views on genetics and race and the inferiority of one race over another. So he got most of his honorary degrees revoked. They're just like, and we don't want you associated with us anymore. So these two guys are putzing around in the lab trying to figure out how to build DNA. The first discovery that helped them out was by a discovery from Chargraff's lab. Chargraff took DNA and realized that the amount of guanine always equals the amount of cytosine, and the amount of adenine always equals the amount of thymine. Every time, every bit of DNA, every chromosome he looked at, same issue. Cytosine always equals guanine, adenine always equals thymine. So if he found that there was 10% adenine, how much thymine must there be? Well done. Now here's where it gets a little bit harder. How much is left total? 80% between the two of those. And if they're equal, how much guanine is there? 40% guanine. And how much cytosine? 40%. That was Chargraff's big contribution. This is where we got the idea that guanine binds to cytosine and thymine binds to adenine. That was big news. Okay, so Watson and Crick now have this new discovery that they can mess around with. G binds to C, T binds to A. But they kept screwing it up. They were binding things and making these tall towers, but they didn't know what the structure looked like. Until Wilkins and Franklin came along. Now, Wilkins was running a lab. Franklin was a postdoc doing her postdoctoral studies, and she was specialized in X-ray crystallography. She would take pictures, to the best of their ability, of proteins so we could look at their structures. And she was going through taking a bunch of different pictures, and she took a picture of DNA. It was crystallized, and she took the picture. It was this picture. Wilkins is kind of a jerk, and didn't really ever contribute anything himself. He took her picture, realized it was of DNA, and then took it over to Watson and Crick's lab and showed it to them and said, hey, look at this picture I took. After he did that, Watson and Crick were automatically like, I got it. I know exactly. This structure, because of the placement of these black dots, shows that DNA is a double helix shape. Well, now they've got a picture of what they're trying to build. They've got the elements. They were able to quickly put it together. They went on to win the Nobel Prize. It was shared by Wilkins, Watson, and Crick, but not Franklin. Unfortunately, Rosalind Franklin died before she was able to even be engaged in the process of getting the Nobel, because you cannot give someone a Nobel Prize posthumously. The men didn't really consider her work that important. In Watson's book, he described her as temperamental, very emotional. Yeah, I know, right? So like, you've been here, right? Very emotional woman. She doesn't belong in a lab. She just doesn't have the temperament. But it turned out without her picture, without her research, her original research, she was the one who was pioneering this field, the discovery of DNA would never happen. So now you know one of the times where science screwed over women. It happens a lot, apparently. Okay, so that was the first science story of today. Nucleic acids. You guys remember nucleic acids? Good, because these are the exact same slides I gave you a while ago. It's the exact same presentation. You experienced this, what, 7 weeks ago? 8 weeks ago? 9? Seems so long ago. Yeah, a long time ago. Right. Nucleic acids are responsible for the storage, expression, and transmission of genetic information. They're made up of monomers called nucleotides. What are the 4 nucleotides in DNA? They're right there, yeah. Adenine, thymine, guanine, cytosine. Thanks for pointing. What about in RNA? What's different? That's right. There's no thymine. There's uracil instead. This blew my mind when I learned it, and I have to tell you guys, it may not be nearly as mind-blowing as it is for you as it is to me, but here we've got an adenine group and 3 phosphates. So what is that called? That's right, adenosine triphosphate. Okay, so far so good. This is so cool. We're good. All right. What if I took off one of those? What's it called? ADP, right? And if I took off another phosphate, now I'm down to 1, now it's called adenine. And that's the nucleotide. I know, that's what I said. It was totally mind-blowing to me, guys. I know you may not appreciate it as much as I do, but that's it. These nucleotides are all made up of a phosphate group, a pentose sugar, and a ring of carbon. Remember, pentose means how many sides? 5 sides. 1, 2, 3, 4. Oh, I lost count. 5. I know, I was only counting to 5. I got a P in there for a phosphate, and then I've got nitrogenous base, these extra rings. That's the nucleotide. There are 2 types of nucleic acids, as we said, RNA and DNA. What's one difference you see? That's right, DNA is a double helix, RNA is a single helix. DNA has what sugar in it? Deoxyribose. RNA has ribose. RNA has uracil, DNA has thymine. And importantly, DNA stores genetic information and RNA decodes genetic information. In DNA, there are 4 bases: adenine, guanine, cytosine, and thymine. They're going to be divided into 2 big classes, the purines and the pyrimidines. Purines are adenine and guanine, and they're larger structures. Pyrimidines are cytosine and thymine, and they're much smaller structures, single ring. Here's how I remember it, and I will expect you to remember it because I'm mean. Purines, I always remember the mnemonic, all pure and good. So adenine starts with an A, like all. Guanine starts with a G, like good. And they are purines, so all good and pure. Adenine and guanine are purines. And as far as the structure, I always remember the smaller word is the bigger structure, because small things have great power. Cool? I will not change the slide because I wanted to also mention there's also uracil. You'll notice that uracil is a single ring. Uracil is a pyrimidine. Purines bind exclusively to pyrimidines. So DNA is double-stranded. You've got all of these nucleotides linked together to each other across the center with hydrogen bonds. What are holding the sugars and the phosphates together? Well, that's a tricky question. So we've got hydrogen bonds going across the center. What's holding the phosphates and the sugars in this backbone together? No, it's not van der Waals forces. Van der Waals forces are very weak, and these are extremely strong. Covalent bonds, yes, covalent bonds. Ooh, and we also learned another term for it, specifically to DNA, phosphodiester bonds. You don't have to remember that one. Covalent, remember covalent. Covalent bonds hold the phosphate to the sugar. Hydrogen bonds hold the 2 strands to each other. Millions of them. Now, one hydrogen bond, very weak, but millions are strong. We're going to need some energy to break that. So, to sum up, differences between DNA and RNA. DNA is double-stranded, RNA is single-stranded. DNA has deoxyribose, RNA has ribose. DNA contains thymine, RNA contains uracil. DNA stores genetic information, RNA decodes genetic information.